Welcome to the Science of Drugs. Today, we're diving into the fascinating world of pharmacology, exploring how medications interact with your body at a molecular level. This is a journey into how a drug goes from a tiny pill to a powerful force for change, focusing on receptors, precise dosing, and essential safety measures. How does a tiny pill actually change your body? The action begins at the smallest possible scale, deep within your cells. It starts when first messengers, like drugs or hormones, bind with their specific receptors to form a drug-receptor complex. This binding relies on an association constant and the drug's intrinsic activity. Once this complex is formed, it triggers the release of intracellular second messengers, such as cyclic AMP, calcium, and inositol triphosphate, which race through the cell to produce the ultimate molecular mechanism of the drug's action. But where exactly does this incredible interaction happen? Understanding the site of action is crucial for designing effective medication. A drug may act in 3 primary locations, depending on its chemical properties and the target structure. Some drugs work entirely extracellularly, meaning they don't even need to cross the cell membrane, such as osmotic diuretics that work on fluid balance. Others act right on the cell surface, locking onto receptors embedded in the membrane, like the antibiotic penicillin or the heart medication digitalis. Finally, certain medications must slip inside the cell completely to work, often interacting directly with DNA, such as anti-cancer drugs and steroid hormones. Next, we turn to one of the most critical aspects of pharmacology, determining the right amount of medication. To figure out exactly how much of a drug you need for an effective treatment, scientists rely on the precise dose-response relationship. When they graph the logarithm of a dose against the biological response, it forms a classic S-shaped or sigmoid curve, which dictates dosing strategy. As the dose increases, the pharmacological response increases in a graded fashion until it hits a ceiling, known as the maximal dose, where adding more drug has no further effect. The exact concentration required to produce 50% of that maximum effect is called the EC50, or ED50, a crucial measure of drug potency. While the dose-response curve focuses on efficacy, the therapeutic index is all about ensuring safety. However, some drug responses are not graded. They are all or none quantal effects, meaning the response either happens or it doesn't. To ensure a drug is safe for the public, scientists must find the median lethal dose, or LD50, which is the exact dose expected to kill 1/2 of a test population. By dividing this lethal dose by the median effective dose, we get the therapeutic index. This index is the definitive assessment of the drug's safety profile. The larger the therapeutic index, the safer the drug is, which is why penicillin is incredibly safe, while digitalis has a very small, dangerous therapeutic window. Understanding these core principles of receptors, dosing curves, and safety indices helps us appreciate the intricate science behind every pill, empowering us to make better decisions for our health. Thank you for watching.